On-the-scene coverage of ACC.13 is supported by Janssen Pharmaceuticals, Incorporated.
Welcome back to ACC.13. We're doing the late-breaking clinical trials. This is On the Scene. I've got a spectacular panel. It's still spectacular, absolutely. So Tony, let me ask you, or Athena, tell me a little bit about SELECT-ACS.
Oh, SELECT-ACS is an interesting trial, Randy. It looks at a new target, and that's P-selectin. And as you know, P-selectin is on endothelial cells, and it's responsible for slowing down the flow of platelets and white blood cells. And ultimately, when VCAM, the other adhesion molecules come into play, then they get stuck to the endothelium, and in fact, platelets release some more P-selectin. So P-selectin is an interesting target, and in SELECT-ACS, they used inclacumab, which is a P-selectin an antagonist inhibitor, monoclonal antibody, and tested the hypothesis that by inhibiting P-selectin, you could reduce the adverse effects of non-STEMIs in patients undergoing PCI. Now, they used a kind of unusual endpoint, which was troponin, but nevertheless, in this study, kind of as a proof of principle that adhesion molecules might be a good target, they were able to show a statistically significant, small but statistically significant reduction of troponin after PCI in these patients. And as you know, there's some evidence that biomarker elevations after PCI augur an adverse prognosis. So some proof of principle of a new agent to attack a new target.
Peter, what do you think?
We always sort of poo-poo troponin and say, well, who really cares? You know, everyone gets a troponin rise after PCI. But you know, it can't be good for you. And so if you can knock down troponin a little bit, I think that's probably not a bad idea. This is unique because it's a new target. And I think we just have to watch and see. The differences are not great, but going forward, it may change the way we think about some of the newer ways of approaching PCIs.
Yeah, I think certainly enough to justify a bigger trial with harder endpoints.
Right. Any adverse effects with its use?
No significant adverse effects, at least at this point in time.
It's given intravenously, isn't it, Tony?
That's right.
1 hour to 24 hours before, so you have some time to give it, and that's a good thing as well.
Yeah, you know, I don't recall that there were significant adverse effects, so let's be clear on that.
So okay, I will be clear. I'll stop right there. Athena, what about TERISA? Tell us about that trial.
Well, TERISA was another trial looking at ranolazine versus placebo in patients with chronic stable angina, and they looked at measuring how often they had frequency of angina.
These were diabetics, is that
These
correct?
were diabetics, that's correct. So it was a different subgroup of patients. Right. And so they had a high rate of anginal episodes, 6.6, 6.8 in the placebo group. And after a run-in phase and 8 weeks out, interestingly, both groups, as we've seen in many of these chronic stable angina trials, had a reduction in their frequency to 3.8 and 4.3. But the ranolazine was statistically significantly better than placebo. Similarly, they measured how often they took nitroglycerin as another marker. They did not do any hard measures, such as putting them on the treadmill and seeing if they developed ischemia. So ranolazine seems to be effective relative to placebo, but again, interestingly, placebo is a pretty good drug.
Peter?
This is an old-time angina trial. You know, it's drug against no drug. And I think I'm going to start a placebo factory and you know, really charge a lot for it because it really works. All you have to do is see your doctor to get better. But we've known that for a long time, and this just proves that point once again. But there does seem to be a little difference, and ranolazine is probably not a terrible drug.
I think the most interesting thing about TERISA is that ranolazine has been shown to have some hypoglycemic effects, and in fact there have been some thought that it might be a good agent for diabetics just for glucose, right, independent of chest pain. And so that's it's a very interesting concept that this was a diabetic study, only in diabetics, and it may be that the drug is a bit more effective in diabetics because of its hypoglycemic
Well, we all
effect.
use ranolazine sort of as a last drug, don't we, down the line after nitrates and calcium channel blockers. This may move it up a notch. It'll be interesting to see.
Yeah.
One of the arguments in using chest pain as an endpoint or symptoms is that they were diabetics who may have a lot of asymptomatic ischemia that we were missing. So.
Well, so it's interesting. So you think either of these, you know, as we look back on the ones we've discussed, all these any of these really sort of strike you as being groundbreaking or, you know, really new developments?
I think the P-selectin business is probably the
yeah.
most groundbreaking. If I had to pick one, I'd pick P-selectin antagonism.
Yeah, I think that P-selectin as a new target is very exciting. Now, I think that there just weren't enough patients in this study to know if there were serious adverse effects. You asked me about that, and in fact, in a couple of instances, the number of adverse events in the inclacumab group were a little bit higher, but the group wasn't big enough for it to reach statistical significance. So we're going to have to wait and see on that.
But I think, as Peter said, and as, you know, Athena, that this is it's an interesting new agent, and you did a great job of explaining exactly what's going on there. So I think that will be exciting. Well, panel's done a good job. You all did a fabulous job, and we hope that you've enjoyed it. We've all learned a lot this morning. We want you to join us again here at ACC 13.